Okay, so we are continuing our conversation about the special senses, and here you can see that somatosensory cortex that we mentioned in class. Let me get my laser pointer here, which you can find here in the parietal lobe right next to that primary motor cortex, which is just anterior. So now we're kind of moving forward and going to take a look at vision. Again, when we think about how this all works, the process of phototransduction is the conversion of light into electrical signals in the rod and cone cells. And we'll talk about the rod and cone cells and the role that they're going to play in terms of transmission of those images up to the occipital lobe. So again, the eyes are composed of an optical component which focuses the visual image on those receptor cells. Again, we said the receptor cells are going to be made up of rods and cones. And then there will also be a neural component which will involve a specific pathway that's going to be a little bit complicated, I guess, that's going to transform those visual images into a pattern and including graded and action potentials that will eventually be perceived then in the occipital lobe. Okay, so when we look at what's going on from an anatomical perspective, things to point out here, you can see ciliary muscles, which are going to be attached both superior and inferiorly to the lens, which you can find here. The pupil is located in the center. We have the cornea, which is superficial and on either sides of the pupus there. Let's see what else. I want to point out the optic nerve is going to exit the back or the posterior side of the eye and allow us to carry those nerve impulses, like we said, to the brain. Let's see, what else do I want to point out? We have vitreous humor, which is in this posterior chamber. Think of this as like a gel-like substance that's going to allow the eye to kind of keep its shape. I think that's probably a good start there. So when we think about how does vision work, one important component that we need to understand is refraction. And refraction is the bending of light. And this is based on the angle at which it strikes the surface of the eye itself and allows us to focus on an accurate image of an object onto the retina. So again, you can see how the light can be manipulated based on how it hits the lens, and that will allow us to stimulate various receptor cells, which are the rods and the cones found on the posterior side of the eye, and allow us to kind of focus in on those particular objects. Okay, and we can actually change our lens essentially. And so we're going to do that through either the contraction or relaxation of these ciliary muscles. And that's going to allow us to do something called accommodation. And accommodation is going to allow us to manipulate that lens in order for us to get things into focus. So again, we can manipulate that lens in order for us to see things up close or far away, depending on what the situation is. Okay, what does this look like? So again, here you can see in focus, we've got relaxed ciliary muscles with a little bit of tension, but not a lot, and light rays from distant objects are nearly parallel, and that will allow us to have a focused image. Okay, if I have those relaxed ciliary muscles when the object is close to me, that's going to create something that's out of focus. And so again, we need to fire those muscles in order for us to manipulate that lens. So we're going to fire parasympathetic nerves and contract those ciliary muscles, which is going to allow us to get that object into focus through the contraction of the ciliary muscles. Okay, so here you can kind of see how this plays out as it relates to nearsightedness and farsightedness. Okay, so again, with normal vision, we're able to get those images in focus regardless of whether they're close to us or far away. Okay, if I'm nearsighted, something that's far away will be out of focus without the manipulation of the lens through glasses or contacts. Okay, versus somebody who's farsighted, something that's up close will be out of focus unless we provide the correction for the lens, whether that's through glasses or contacts. So again, kind of opposite issues when we think about what's going on with the ciliary muscles when we look at nearsighted versus farsighted individuals. Okay, so as we mentioned, we have these photoreceptors that are going to be found on the posterior internal portion of the eye, and these photoreceptors are going to be made up of rods and cones, and they are going to work in cohesion to essentially grab the light and allow us to create those images, recreate those images within the occipital lobe. And again, you can kind of see here the cones look a little bit shorter in comparison to the rods are a little bit longer. Yeah. So when we think about what this looks like from a neural perspective, again, those receptors or those photoreceptors are going to be important for us in terms of allowing us to send that signal through this pathway that we're going to talk about here on the next slide. But again, we think about how these kind of relate to other functions. Certainly we're receiving, light versus dark information, and that's going to play into the biological clock and kind of have an influence in some of these other areas. In particular, it will be important in determining our circadian rhythm based on, those sleep wake cycles or the light dark cycles that we are kind of exposed to. That information is coming from these photoreceptors, but isn't necessarily directly like going to that vision center within the occipital lobe, but it's going to go to some other areas to allow us to kind of play around with melanin in terms of creating feelings of sleepiness versus like arousal. Okay, so let's take a look at the neural pathways associated with vision. So again, first things first, we're going to have photoreceptor stimulation. So again, we're going to activate the rods and cones based on those wavelengths of light that are reflected off of the lens. And those photoreceptors are going to then interact with the bipolar cells. And bipolar cells are then going to interact with ganglion cells. Okay. Then those ganglion cells are going to cross over what's called the optic chiasm. And the optic chiasm, think of it as like a cross where I'm going to take the info from the left and switch it over to the right side and vice versa. Then we'll move into the lateral geniculate nucleus, and this is where those optic nerves are going to terminate. This is within the thalamus, and then that information, as we know, the thalamus is going to kind of serve as like a relay station for that sensory information. The thalamus is then going to take that information and send it over to the visual cortex, which is found in the occipital lobe, and that's where we will eventually perceive that image. Let's see, other things to note, the lateral geniculate nucleus will also send information to the brain dealing with those biological rhythms, including the epithalamus and the pineal gland, where we are going to deal with melatonin, which again we said is going to be important for like the sleep-wake cycle. So again, that's kind of one of the reasons why they say like blue light and being on your phone or watching TV or whatever right before you go to bed can kind of stimulate you to stay awake and alert and can affect your sleep quality. And that's because of this system. Okay, so again, if I'm receiving information about that light and that stimulation within the lateral geniculate nucleus, then it's going to maybe affect the release of the melatonin and our sleep-wake cycle. So again, that's one of the reasons why you're supposed to kind of like try to get calmed down and, bring the lights down, not be on your phones or watch TV like right before you go to bed or within like those last 30 minutes before you go to bed. And again, the reason for that is it's supposed to improve your sleep quality. Let's see other information about these visual pathways. Other visual information is going to pass to the brainstem and the cerebellum, and this will be used for the coordination of our eye and head movements, fixation of gaze. So again, being able to kind of manipulate the lens, whether we're trying to focus on things that are close or things that are far away. And then change in pupil size, again, that can be manipulated by how bright it is as well, right? So again, the dilation or the like constriction of your pupils can vary kind of based on the amount of light that we're experiencing. Okay. This is just a nice image that kind of shows you what's going on here. So again, we said that we're going to start with those photoreceptors on the interior and posterior side of the eye, and those are the rods and cone cells. Then we're going to travel through bipolar cells to ganglion cells. Then we're going to reach the optic chiasm, where we're going to have that information switch over from left to right. So it's again creating that crossover effect. Okay. And then we will go and terminate in the lateral geniculate nucleus, and then that information will eventually travel to the occipital lobe, to that visual cortex. Okay, so again, we said that there are a couple different types of cells. Okay, so we have rods and cones, but in reality, we have three different types of cones. We have short wave cones, medium wave cones, and long wave cones. And then you can see the rods, how they kind of look different. Again, these receptor cells are named kind of based on their visual appearance. So again, the cones have this kind of tip, and then the rods look more like a cylinder. Okay. Let's see. What do I want to tell you about these? In order to discriminate color, this will depend on the intensity of the light striking the retina. So brightly lit conditions, the differential response of the cones allows for good color vision. And in dim light, the rods will be able to respond. Okay, so the rods are going to be better, like in dim light situations. The cones are going to be able to respond to high color types of things. What else? The rods are also going to be better at perceiving like quick movements. Again, think of these as like being important for survival. So again, if it's dark and there's a predator, or I'm worried about being chased or something along those lines, I need to be aware of quick movements. And I'm not so concerned with like what color the tiger or lion is. I'm more concerned with their movements and where their general location is. So again, less emphasis on like the I guess strength of the like colors, and more emphasis on detecting movement and seeing things in shades of gray that are maybe like in a darker lit environment. Okay, color blindness, I just thought this was interesting, but there's several types of defects in color vision, which generally results from mutations in these cone pigments. And the most common form of color blindness is the red-green color blindness. And this is predominantly found in men, affecting 1 out of 12, which I thought is just nuts. Like, that's way more than I thought. So men with red-green color blindness can either lack the red or green cone pigments entirely, or they have them in an abnormal form, which kind of results in those colors kind of looking like grayish or like the same kind of. So again, they can't necessarily differentiate between red and green, which is why like our stoplights are set up the same. So even though, you have green, yellow, red, they're always posted in the same like positioning for people that are colorblind, so that they know, okay, the top one means this, the bottom one means this. So again, they kind of learn that in a little bit different of a way, just because of not being able to differentiate those two colors. This just kind of shows you what this might kind of look like. So again, non-color blind range. So here you can see, your purple, your blue, your green, your yellow, your reds. If our red cones were defective, this is, I think, kind of cool. It just shows you what they see instead. So instead of seeing like this purple, which would be like your red and blue, you just see blue. And instead of seeing that true red, it's more like a gray. Okay. So again, if you had the defect in green cones, so again, same gig with the green here. The green just kind of looks like gray in comparison to, what the average person would see as kind of like a true green. Okay. When we think about eye movement, We have 6 skeletal muscles that are going to attach on the outside of the eye and perform two types of movements. Okay, we have the 2 types of movements. We have slow and fast. Okay, so the fast movements are saccades, and saccades are going to be fast, jerking movements that bring the eye quickly to one fixation point. Okay, so again, it's going to allow us to quickly respond and be able to get into focus for something that's, maybe moving in our periphery quickly that is unexpected or something along those lines. And then our slow types of movements. So we have slow eye movements that are involved in both visually tracking objects. So again, if I'm watching somebody walk, I'm able to kind of manipulate my eyes across my visual scanning in order to kind of keep my eyes on them as they move. And also during compensation for movements of the head. So maybe I want to keep watching them as I'm moving my head this way as I'm walking or something along those lines. So again, those slow movements, we're going to have particular muscles that are involved there. Okay, so that gives us a little bit of information about the visual paths and kind of how that works. And now we're going to move forward and take a look at hearing. Okay, so hearing, sense of hearing is based on the physics of sound and the physiology of the external, middle, and inner ears. So we kind of have 3 different sections there. And how does this work? We're going to have sound transmission through sound waves that will be converted to vibrations, and then they're going to stimulate specific mechanoreceptors, which we'll talk about here shortly. Okay. So how does this work? So again, we're going to create sound waves when we talk or when we hear music or any type of sound. And these are kind of like pressure waves. And these waves can vary in amplitude and frequency. Okay, so amplitude is like the height of those. And that's going to give us like different loudness. So again, the height or the amplitude of these waves determines how loud someone is. And then the frequency of those waves will determine the pitch. So if it's high pitched versus low pitched, so high pitched is going to have a high frequency and low pitch is going to have more like a low frequency. Okay, so let's take a look at all of these components here. Okay, so here you can see the external ear. And the first portion of this is known as the auricle or the pinna. And that is just like the external elastic cartilage that we can find here. And this is going to act like a funnel. Okay, so the external portion of our ear will act like a funnel that will allow those sound waves to travel into the external auditory canal. And as those sound waves travel through the external auditory canal, they're going to reach the external acoustic meatus, which would be kind of like right at the edge here. And the sound waves are going to travel through here. Okay, so they'll come through the canal. And then, I'm sorry, I said that wrong. So you have the pinna, then you have the external auditory canal, and the external auditory meatus is the same thing. My apologies. Okay, so that's this purple section. And then you're going to reach the tympanic membrane, which is kind of where those sound waves are going to stop and be changed. And this is also known as the eardrum. So the tympanic membrane, also called the eardrum, and this is where those sound waves are going to be transferred into vibrations. And this is going to happen due to a change in pressure. Okay, so again, to kind of summarize that, the sound waves will be collected by the pinna or the auricle, which is the external portion of the ear. Then it will travel through the external auditory canal or the external auditory meatus. Then it will reach the tympanic membrane, also known as the eardrum. And this is where those sound waves are going to be changed from waves into vibrations as we move next into the middle ear. Okay. In the middle ear, we're going to have the malleus, incus, and stapes. Okay, so you can see those 3 kind of in this green section here. And then the next thing that's going to happen is that these vibrations are going to be able to move through this oval window or the stapes here into the internal ear, which in our conversation, we're concerned with the cochlea. And the cochlea will kind of be the last portion of the sensory process before we start sending nerve impulses to that auditory cortex within the brain. Okay, so that kind of gives us an overview of what's going on. Let's zoom in and take a look at what's going on here in the green with the middle ear section. Okay, so here you can see that external auditory canal and the tympanic membrane or the eardrum. Okay, and then we're going to move into this green section, which is known as that middle ear, also sometimes called the tympanic cavity. Okay, and as we said, it's going to contain 3 bones. It's got the malleus, incus, and stapes. And these are also known as auditory ossicles. And basically their job is to like make the vibrations more like what's the word I want to use, like increase their amplitude, make them like more prominent basically, so that when we convert them down into the cochlea, they're like a little bit more prominent basically. Okay, so the inner ear, which you can see here in blue, we've got a couple different things going on within the inner ear. We have the labyrinth, which is filled with fluid, and that's going to be part of our equilibrium system. And then we also have 3 semicircular canals, which are going to be important for that vestibular system as well. So they're in charge of balance and things like that. And then we have the cochlea, which is kind of the last portion of this hearing process. Okay. Entrance to the cochlea is going to be this round window, which you can see here. And then we have receptor cells in the cochlea that have hairs. And as these vibrations cross over those hairs, this is going to cause the stimulation of these sensory receptors, and these specialized mechanoreceptors, known as those hair cells, are then able to do something with those vibrations or with those the sound that's coming through. Okay. So here you can kind of see what's happening here. So we have those middle ear bones, and they are going to like amplify the vibrations. And then we said the vibrations are going to transfer through the round window. As they move through the round window, they're going to stimulate the hair cells or these specialized mechanoreceptors, which are found in the cochlea, and that's going to allow us to receive that information. Nice little chart here. Again, please pay attention to the arrows. They're not very large, so my apologies there. But this just kind of summarizes what's happening from an auditory perspective. So number 1, sound waves enter that external acoustic meatus. The waves are going to produce vibrations on the eardrum or that tympanic membrane. Then those vibrations are going to transmit through the 3 middle ear bones, so the malleus, incus, and stapes. Okay. Then next, those vibrations are going to travel through the round window into the cochlear duct, and the vibrations cause stimulation of those hair cells in the cochlea. And again, those are specialized mechanoreceptors, which is going to allow us to then send sensory information via sensory neurons to the temporal lobe of the cerebral cortex, which is where we find that auditory cortex. Yeah. Okay. So next, let's take a look at what's happening within this vestibular system. Again, we said that within the inner ear, we've got 3 different like little semicircular canals. We have the vestibule, utricle, and saccule. Okay, and let's see. So you can see saccule is here, utricle here, and which one am I missing? Vestibule. Vestibule here. Okay, and then you have these 3 semicircular canals. Okay, the vestibule, utricle, and saccule are going to be in charge of like static equilibrium or maintaining our balance when we're kind of in a resting position. And then those semicircular canals are in charge of maintaining balance during movement. Okay, so those are dynamic in nature, and those are going to have a specific fluid inside of them that's going to kind of move around when activated, and that's going to assist us with maintaining our equilibrium. Okay, so how does this kind of work? So these semicircular canals are going to contain sensory hair cells, and they're going to sense rapid changes in head position. Okay, so again, that's kind of how they work. And if we take a look within these semicircular canals, there's a cupula and an ampulla, and you can kind of see those here. And again, those hair cells are going to be stimulated by the change in that fluid. Okay, so again, as I like rotate my head or whatever, that fluid's going to kind of swoosh around, and it's going to manipulate those hair cells and activate them to allow us to understand that there's been a change in body position and allow us to kind of maintain our balance in that way. Okay, so here you can kind of see what's happening within these hair cells. So during resting activity, you can see that we're receiving some feedback, but not a lot. And then as we start to stimulate them because we're creating a spinning motion or whatever, the depolarization rate is going to increase in frequency. And then you can see inhibition or hyperpolarization occurring as well. So again, imagine like we spun around in a chair, and again, we're going to be stimulating those hair cells as you like spin around in your chair. And then if we were to stop immediately, your eyes would still be like ticking and you would feel dizzy when you stopped. And the reason that you're going to feel dizzy when you stop is because of this hyperpolarization. Like even though your body has stopped, the fluid within those systems is still kind of swirling around. And so therefore, we're still experiencing some action potentials, which is why we feel dizzy for a little bit until that fluid kind of like settles down. Okay, here you can see the relationship between the utricle and saccule. Again, these are going to be important for static. And so if you've ever like bent down, tied your shoe, and then tried to stand up and felt like, whoa, like a little bit dizzy, again, that could be that hyperpolarization of those hair cells on your return back up to a standing position. These vestibular pathways or this vestibular information, we use this for a lot of different things, but essentially it's going to help us to control eye movement. It's going to help us to maintain posture and balance and provide awareness of body position and acceleration as well. And interestingly, like the fluid inside of these tubes gets like thicker as you age. So I think this is super interesting, but like when I was little, like I could go on, swing, water slide, roller coaster, no problem. Any of that stuff didn't give me any issues. And now it's like I get on the swing with my kids and I feel like dizzy, like I'm gonna throw up. Which is just nuts to think like that fluid just becomes thicker and less responsive as you age, which is why like kids don't have any problems with doing like a lot of those like roller coasters and that type of stuff versus when you reach adulthood or even like later in life, as that fluid becomes thicker, it's like less responsive, or the vestibular system, I would say, is not as not as responsive. Yeah, we could say that. And so therefore, like we become dizzy easily and stuff like that, which is annoying because I want to be fun, and it's like I feel like I'm gonna throw up if I try to do the swing set, and so it's like, what the heck, people? Okay, so again, when we look at this pathway, what's going on here, to kind of summarize. So again, the hair cells are going to be activated when that fluid kind of pushes over the top of them. Again, mechanoreceptors, they'll be depolarized, and then that information will transfer to the vestibular nerve, and that information will go to the brainstem, and it'll be integrated at the level of the thalamus and parietal lobe so that we can kind of decide what to do. So we do have a vestibular center that's located in the parietal lobe. And again, that vestibular information will also be integrated with information from like the proprioceptors that would tell us information about muscle, joint, bone positioning, and things like that, which would kind of allow us to have a feeling of control over body positioning and allow us to manipulate that if we lack that control. Okay, so that's a little bit about the ear and about the vestibular system. And now we're going to move forward and take a look at these chemical senses. So we're going to take a look at taste and smell. And these are going to be perceived through chemoreceptors. And again, they're closely linked, as you might be aware, and are going to have some very close similarities when we think about how we're going to process this information. So let's start with taste. Taste or gustation works via the taste buds, and taste buds are like a small group of cells arranged. Think of it like an orange slice. And basically, you have microvilli that are going to be sitting kind of on top of these taste buds. And this is important because the more surface area I have, the greater the stimulation can be. And so therefore, I can differentiate and experience more tastes or a great variety. Okay. So let's see what else. At the bottom of the taste buds, we have basal cells, which are going to divide and differentiate continually. So again, an area of high metabolic activity because of the harsh environment in the mouth. Again, think about like the acidic foods that you might consume, ketchup, for example, or spicy things like buffalo sauce or something with a great amount of abrasion, like Doritos, for example. So again, the mouth is a very harsh place to live. So we've got a very high metabolic rate here. Okay. So how does this work? To enter the pores of the taste buds and come in contact with those taste receptor cells, foods must be dissolved in liquid. So again, if we're try to eat like a dry cracker, we're not going to be able to absorb the taste without creating any saliva. Okay, so again, we either have to like consume that with a swig of water or something like that, or we have to have some secretion from those salivary glands in order for us to be able to perceive those tastes. So I think this is kind of interesting. So many different chemicals can generate the sensation of taste by differentiating a few basic types of receptors. So we have sweet, sour, salty, bitter, and something called umami, which is a Japanese word that means delicious. And basically the umami, think of it as like almost like a savory steak or something like that. So again, think of like a meaty, nice protein, something along those lines, and that's kind of that umami flavoring. What's cool about our taste buds and the different variety of foods that we consume is we can activate these receptors in so many different combinations, which is the spice of life, right? So again, being able to understand how we can kind of activate these in various ways based on what it is that we're consuming, I think it's fun. It's pretty cool. We also have like when we eat spicy foods, I can't remember if I have this on one of the slides coming up or not, but when we eat spicy foods, we're actually activating nociceptors and thermoreceptors, which is why, like, if you were to eat like a jalapeno pepper or something and you start sweating in here, up here, that's because you've activated those thermoreceptors. And like, what a bunch of weirdos. We're activating nociceptors when we eat because of that spicy food. But again, I like spicy, so I'm fine with it. But I'm also, I'm a little odd, so it's fine. I don't know. Some researchers believe that your taste buds are going to be more like um, there's like a greater abundance of these particular types of taste buds in these various locations. That's not to say that at the front you don't also have salty, sour, and bitter. You just have more sweet here than in other locations. So again, they think that's probably how the tongue is kind of divided up. Again, people are going to vary in terms of probably how many of each receptors they have. If you have a big sweet tooth, maybe your sweet section is a little bit larger, or maybe it has to do with the release of those kind of like those feel good hormones associated with eating something sweet. Versus some people would rather have steak and potatoes or something more like savory like that. Okay, well, maybe your salty section is larger and your sweet section is smaller, or maybe it has something to do with that processing once we've consumed that in terms of the satisfaction piece. So, not really like 100% sure how all of that works. But again, we do have, like I said, some pain receptors kind of mixed in here as well. So, like chili peppers and ginger might activate those pain receptors. And while, like I said, while all receptors are located all over the tongue, there is a good amount of thought that certain types are more concentrated in certain areas. So again, when we think about how does this work in terms of signaling those taste receptors, each group of tastes has a distinct signal transduction mechanism. So salts, the salt taste will be related to, not surprisingly, sodium ions. Okay, so we're going to use sodium ions for that transduction mechanism. Sour taste is going to use hydrogen ions. Not sweat taste, but sweet taste will come from glucose or G protein coupled molecules. Okay, so the sweet taste will come from like glucose, not surprising as well. Bitter flavor is associated with many poisonous substances like plant alkaloids and arsenic, and this is going to be G protein coupled. And then umami receptor cells can depolarize via a G-coupled protein receptor mechanism as well, right? So again, just really kind of interesting when we think about how we are perceiving each of these in kind of a different way. Okay, so what about the olfactory receptors? Again, these are also a chemoreceptor that's going to be kind of specialized, but the olfactory receptor neurons lie in the olfactory epithelium, or the upper part of the nasal cavity. And these only last about 2 months, and then we're going to replace them pretty frequently as well. And again, what's going on here? We've got some cilia that will contain receptor proteins that provide binding for those odor molecules. And then the axons of the neurons from the olfactory nerve will receive that sensory information and allow us to transfer it to the brain. And that is cranial nerve 1, or that olfactory nerve. The proteins in the mucus may also interact with the odorant molecules and transport them to the receptors, which facilitate the binding to the chemoreceptors. So again, some mucus is going to be helpful for us in terms of receiving smell, but too much would block some of these receptors, which is why when you have a cold and you're like very mucusy, you're not able to smell things well. And again, because these chemoreceptors are so closely linked with our sense of taste, because those pathways are going to be very comparable when we take that information up to the brain. That's why when you have a cold, food doesn't really taste as good as it would when you can also enjoy the smell of that food. And then lastly, when we think about olfactory or factors that affect our sense of smell, the discrimination can vary based on our attentiveness. So if we're paying attention, we want to smell the wine or we want to smell those cookies, that can make a difference. Hunger. So again, sensitivity is greatly affected by hunger. So again, if I'm hungry, man, everything smells good, right? So again, that can play a huge role. Gender, so women generally have a better sensitivity to their olfactory sensations in comparison to men. Smoking will decrease our sensitivity. Age, so again, as we age, our sensitivity will decrease as well. And then state of olfactory mucosa. As I mentioned before, if we have too much mucus, this will decrease our ability to receive senses. And there is actually a genetic defect where they can't smell anything, and that's so sad. Onosmia is what that's called, when I'm not able to perceive, or something is wrong with those chemical pathways to perceive smell. All right, I think that's it for today. I hope you have a great day, and I will see you soon.